Hello, I'm Joanna Shields, CEO of BenevolentAI. I'm excited to share our progress with you today and demonstrate how the BenevolentAI technology platform is driving transformation in drug discovery and development. But before I begin, I'd like to draw your attention to the disclaimer notification on the screen. I joined Benevolent nearly 5 years ago to align emerging technologies with science in a bold new way. And today, Benevolent is a clinical stage company with a full suite of AI augmented intelligence tools that support scientists in discovering novel targets and progressing them into the clinic. We focus on complex multifactorial diseases with high unmet need, and our platform works in any therapeutic area or modality. And what makes BenevolentAI unique is that our technology platform has generated every asset in our pipeline, which now includes 15 named drug programs and 10 exploratory stage programs. Our most advanced asset is for atopic dermatitis, which is currently in phase 2, with data expected in the first quarter of this year. Our next most advanced program is a very exciting novel asset that we discovered and we are developing for ulcerative colitis. We filed the clinical trial application in December and expect to start a phase 1 study in the first half of this year. Our highly productive collaboration with AstraZeneca is an important revenue driver for us and provides strong scientific and commercial validation, with 5 novel targets already selected for AstraZeneca's portfolio. I'll explore these points in more detail later in the presentation. Now let's turn to the problem that we set out to solve. Today, biomedical data and research is growing exponentially, and as humans, we can't possibly keep up. But with the help of AI and machine learning to augment human intelligence, we can. When referring to this information paradox, Nobel laureate Sidney Brenner, he said, "We are drowning in a sea of data, yet we're starving for knowledge." At Benevolent, we set out to make this vast and expanding universe of biomedical data and research accessible and useful for scientific inquiry. Our AI discovery platform and machine learning tools empower scientists to discover new knowledge, accelerate innovation, and increase the probability of discovering a successful drug. So, how do we do this? We built a robust data architecture and knowledge graph that captures a multi-dimensional representation of human biology across all diseases, opening up the entire disease landscape for exploration. Our methodology is unique in its scope and ambition, and we deliberately choose not to restrain ourselves to any one data type, therapeutic area, modality, or experimental methodology, all of which have limitations and biases. We ingest and normalize data from diverse and independent sources and tens of millions of peer-reviewed research papers, combining all that with proprietary data that we generate from the experiments we run in our labs. Our machine learning models reason across all this data, extract insights, and infer millions of connections, describing how biomedical processes interact in a human system. This approach is game-changing to understanding complex multifactorial diseases where the underlying causes are misunderstood. Using our multi-omics pipeline, we integrate patient data and insights from transcriptomics, genetics, and genomics. We generate endotype-specific target predictions that maximize our chance of clinical success. Finally, our data architecture is built for scale and flexibility, so we can rapidly capitalize on valuable new data sets as they become available. Now let's turn to our platform and suite of AI tools that allow scientists to gain a comprehensive understanding of biological effects, refine hypotheses, and predict high-confidence targets. By deciphering complex disease biology at the earliest phase of R&D, we believe that our AI platform enables better decisions and increases the probability of selecting the right drug target at the outset, reducing costly failures down the line. Now, I would like to walk you through how our tools navigate these disease networks, formulate relevant biological questions, and generate target predictions. To demonstrate this, let's look at ALS as an example. Here, as we explore the disease network and biology, our algorithms surface the top-ranked mechanisms associated with ALS, such as RNA localization and autophagy, and also display useful information on how much data exists relating to this condition, the mechanisms involved, and their relevance to the disease. The tool visualizes the interconnectivity of biological entities and processes and how they interact in a human system. In this highlighted example, denoting a signal and directionality, in this case that SOD1 is downregulated and that it acts on DERLIN1. Each query allows scientists to interrogate the disease network and the underlying data supporting the biological mechanisms surfaced by the tool. Our platform supports how scientists work, allowing fast exploration of disease biology in unprecedented detail, and surfacing potential targets and mechanisms they may not have considered otherwise. At the end of this process, we have a multifactorial question ready to feed into our predictive models, guiding the model to uncover novel targets and rank them according to the criteria set by the scientists. An example of this is shown in the blue box. In this case, the question posed is, can we treat ALS by reversing autophagy impairment in microglia by reducing oxidative stress? Next, we run our suite of machine learning prediction tools optimized to point scientists towards new knowledge and ultimately novel discoveries. Our AI models learn and generate proprietary knowledge from the vast corpus of biomedical literature in our graph, and then propose targets by ranking the entire genome from the most relevant to the least likely involved. These large language models are purpose-built to augment critical R&D decisions and predict drug targets, whilst being evaluated against drug discovery benchmarks, fine-tuned on internal proprietary data, and fully integrated into our tech stack to overcome potential limitations. This powerful suite of AI tools and models collectively expands the universe of drug targets, and most importantly, novel targets that have never been before associated or considered for a disease. And finally, our platform is modality agnostic, so it can predict targets that can be drugged by antibodies, small molecules, and other approaches. Now let's focus on the triage process. Running these powerful predictive models generates ranked results ready to be assessed by scientists. The next suite of tools automatically filters vast quantities of data underlying target predictions to determine progressibility, selecting only the most promising targets to take into wet lab experiments and to mitigate the downside risks and later stage failures. Here, we have a view of what this looks like. The system generates a ranked list of predicted drug targets, each representing associated factors and evidence to support the prediction. Again, we're looking at ALS. Note that these targets are arranged and color-coded by their likelihood to fit a predefined disease program strategy. In debilitating diseases like ALS, tolerance for side effects may be quite different from non-threatening diseases. Therefore, scientists must be able to optimize for what they determine are the most critical factors and systematically triage the predicted targets to determine which ones are best to test in an experimental assay. Now let's look at the evidence the system provides. The assessment criteria along the left side, such as biological rationale, ligandability, safety, druggability, and therapeutic evidence, can be customized or prioritized to fit the disease hypothesis. Then, by clicking on the Literature tab, our large language models rank billions of sentences from tens of millions of documents in our graph to extract and rank the most relevant evidence to support the target predictions. The evidence presented represents the model's reasoning and helps scientists decide whether the inferential leap is grounded in biological fact, helping them trust the outputs and ensuring our approach is not susceptible to plausible sounding but ultimately false inferences observed in open source alternatives. As our language model training is integrated with our knowledge graph pipeline, it helps us uncover literature evidence that we would not have found otherwise through traditional search approaches or keyword matching. This whole system was co-developed by our technologists and scientists to provide them with the information they need to triage possible targets, all in one carefully optimized view. Generating these critical insights at this speed and scale to support and guide R&D decisions is incredibly valuable and illustrates the power of our augmented intelligence approach. Selected targets then pass through rigorous experimental validation at our state-of-the-art wet lab in Cambridge in the UK. We continue to invest in cutting-edge methodologies, including CRISPR, RNA-seq, and human iPSCs, to evaluate target hypotheses in cell-based models. Finally, the more we do, the more we learn. We use these experimental insights to enrich our graph and enhance future target predictions in a systematic learning loop. As I've demonstrated, our approach enhances drug discovery in several distinct ways. First, by expanding the universe of possible drug targets. Our comprehensive data foundations and AI models are optimized to surface novel targets across any therapeutic area or drug modality. Second, our platform is built for scale and can support multiple in-house and partner deployments simultaneously. And by integrating our platform alongside our AI chemistry capabilities and our wet lab capabilities, we can significantly reduce drug discovery and development timelines, ultimately increasing the probability of success down the line. So, how is this all translated into impact? It's best represented in the productivity of our platform and discovery engine. This chart by LEK analyzes the number of IND filings per year for major pharma and biotech companies. It shows the potential for BenevolentAI's output to match the R&D productivity of much larger companies at a fraction of the cost. So now let's turn to the business model. Our platform unlocks multiple routes to value creation. We can rapidly move in-house programs from discovery to preclinical and clinical development and explore strategic licensing, development, and commercialization partnerships at the optimum stage for each asset in our pipeline. We also have the potential to scale our platform in pharma partner environments, as we have with AstraZeneca, and to unleash the full potential of our technology through non-commercial collaborations. So let me show you our progress in each of these areas. First, beginning with our pipeline. We have built a substantial in-house pipeline with a healthy balance of best-in-class and first-in-class programs, spanning multiple therapeutic areas. Our growing pipeline reinforces our end-to-end discovery and development capabilities. All of our programs have been generated and developed in-house using the Benevolent Platform, or in the case of CKD and IPF, in partnership with AstraZeneca. Although our platform can discover novel targets in any disease area, as our pipeline matures, we will focus internally on 3 core areas for in-house development. These include immunology, neurology, and oncology. We continue to have a small number of existing programs outside these therapy areas, which we will look to co-develop or license at the earliest and optimum time. Our most advanced asset is BEN-2293, a potent and selective inhibitor of the 3 TRK receptors, which we discovered and developed at BenevolentAI for atopic dermatitis, and is formulated to be administered topically. There is a clear unmet need in atopic dermatitis for a topical drug that addresses itch and inflammation in mild to moderate patients with fewer side effects than current treatment options. We successfully completed the Phase 1b safety and tolerability study in 32 adults with mild to moderate disease in December 2021, which, as you can see from the turquoise line on the screen, demonstrated a trend towards a clinically meaningful effect. However, it is important to note that this part of the study was not powered to assess efficacy. We embarked on a Phase 2a study in 90 patients last year, and we expect data in the first quarter of 2023. With this data package, we intend to out-license the asset for onward clinical development. Our next most advanced program is BenevolentAI, a novel PDE10 inhibitor under development for ulcerative colitis, a disease with high unmet need, with 40% of patients not responding adequately to anti-TNFs or who have become refractory to care within a couple of years. There are also severe safety deficits in current therapies, with agents such as JAK inhibitors carrying black box warnings. Consequently, there is a clear opportunity to differentiate through the delivery of an oral, safe, and efficacious treatment for moderate to severe patients. As part of the rigorous process of validating this hypothesis, our academic partner, Tom MacDonald, professor of immunology at Queen Mary University in London, tested a representative PDE10 inhibitor on surgical colonic biopsies taken from UC patients that were refractory to standard of care therapies, which is the precise patient group we will be initially targeting. As you can see in the bar graph, the PDE10 inhibitor was able to demonstrate a significant reduction in the inflammatory cytokine markers, IL-6 and IL-8, relative to the controls. This is the first ever demonstration of a PDE10 inhibitor showing efficacy in a UC system, with a compelling data package to take forward into clinical development. The Benevolent Platform uncovered PDE10 as an intriguing target for UC, despite the fact that it was entirely novel, with no known research linking it to the disease. Our chemistry team then designed and developed a potent, selective, and peripherally restricted candidate molecule within 2 years. This asset, BenevolentAI, has transitioned through Reg Tox, and we submitted a clinical trial application in December 2022. We look forward to commencing first-in-human studies within the first half of this year. In support of the program, our precision medicine team are using patient-derived molecular descriptors to identify optimum patient cohorts to inform our clinical trial design and identify efficacy biomarkers to further increase our probability of clinical success. In the interest of time, I've only touched on our 2 most advanced assets in our pipeline, but we have a robust pipeline of programs across a range of therapy areas, all again generated by our AI platform and molecular design capabilities, which have enabled us to rapidly build a portfolio of targets with a substantially projected market value. Turning now to our successful collaboration with AstraZeneca, who've already validated and selected a total of 5 novel targets identified by our platform for their drug development portfolio in 2 disease areas, IPF and CKD. As part of an expansion of this collaboration announced early last year, work is underway in 2 new therapy areas, heart failure and lupus, for which our platform has already identified potential targets that are undergoing experimental validation by AstraZeneca. This is a unique partnership, a coming together that combines AstraZeneca's world-leading disease expertise with BenevolentAI's advanced AI and machine learning, and is indicative of the potential to scale our platform and unlock new routes for value creation. Now turning to how we use AI for wider societal benefit. Benevolent is a purposeful company, and we believe it is important to amplify the impact of our platform. One specific example is how during the early stages of the pandemic, we deployed an AI workflow to identify any drugs that could possibly be repurposed to treat COVID-19. Our technology determined that baricitinib, an approved rheumatoid arthritis drug owned by Eli Lilly, had an unknown antiviral mechanism that would make it a strong treatment candidate, and we published our research in The Lancet February 4th, 2020. Fast forward to today, that hypothesis continues to hold true. Baricitinib is now fully approved by the FDA and other leading regulatory bodies to treat COVID-19 and has been shown to reduce mortality in multiple randomized clinical control trials, including Eli Lilly's COVE-BARIO trial, which showed the drug reduced deaths by 38% in hospitalized patients, rising to 46% for those on supplemental oxygen. Identifying this drug so quickly is a testament to the power of our platform and its potential to enhance and accelerate life-saving research. Using our same AI workflows, we initiated a research collaboration with Drugs for Neglected Diseases in Switzerland, which aims to deliver biological targets and drug repurposing candidates to treat dengue fever, a significant healthcare burden in the developing world. These non-commercial collaborations demonstrate our commitment to using our platform for broader societal benefit. At the core of BenevolentAI is a world-class team of scientists and technologists who work side by side to co-create our platform and discover and develop life-changing new treatments. We are supported by a renowned board of industry luminaries, led by our chair, Dr. Francois Nader, a veteran biopharma executive who also serves on the board of Moderna, and his independent directors including Dr. Olivier Brandicourt, the former CEO of Sanofi, Dr. John Orloff, former head of R&D at Alexion, and Sir Nigel Shadbolt, principal at Jesus College at Oxford University and founder of the Open Data Institute. Our business combination with Odyssey acquisition in April last year strengthened our financial position with a cash runway to Q4 2024, providing sufficient capital to support the clinical development of our in-house pipeline assets and drive long-term value creation through out-licensing over the next 1 to 3 years. We expect the full data package from the phase 2 clinical trial of our asset for atopic dermatitis in Q1 this year, so by the end of this quarter, which we intend to out-license for onward clinical development. We also expect to begin the phase 1 study for BEN-8744 as a first-in-class treatment for ulcerative colitis in the first half of this year. You can see from our growing pipeline how the Benevolent Discovery Engine can consistently generate valuable drug programs and will continue to build our portfolio, aiming for a healthy balance of first-in-class and best-in-class assets, with 1 to 2 clinical trial applications or IND stage drug candidates every year. We also aim to scale our platform further with additional pharma partnerships that leverage our disease-agnostic capabilities to generate incremental value. The progress at BenevolentAI demonstrates that AI adoption is already unlocking the next wave of biopharma innovation. With our substantial portfolio of platform-generated drugs, our work with pharma and research collaborators, and our continued investment in state-of-the-art technology, we're showing every day how AI can be used to decipher complex disease biology, uncover new knowledge, and enhance scientific discovery. We believe that our platform and AI tools need to be shared more, and our ambition is to put them in the hands of thousands of scientists to unlock the mysteries of disease and facilitate the scaled development of new, more effective treatments for the patients who need them. This is why the work that we do matters, and I'm pleased to invite you all to partner with us and join us on this journey. Thank you.